Quick question, how many of you know the difference between analog and digital? Google it if you have to. When we talk about the nervous system, this is a big deal. The world comes at us in an analog way, smells, sounds, light. But for us to understand it, for us to comprehend it, we have to turn it into a digital signal. In looking at the nervous system, what allows you to click a mouse, what allows you to blink, what allows you to hold a pencil, what allows you to fall in love, what allows you to have fears, have nightmares, have great dreams, it's all about digital. That's how the nervous system works. Just to kind of point this out and put this up front, we could do 16 weeks of neurophysiology easily and not even scratch the surface. So what I want to be able to do, because we don't have 16 weeks, is to give you an understanding of how essentially the nervous system works. So here's a kind of comparison between analog and digital. Analog is fluid. Back in the day when we used to listen to albums, the sound coming off of the album was fluid, it was changing, it was fluctuating up and down and up and down. Today, most of you, if not all of you, live in a digital world. A 1 or a 0, it's binary. Is it on or is it off? You stick a CD in, you stick a thumb drive in, it's all digital. So what does this have to do with the nervous system? Well, here's a couple of examples to make a couple of comparisons. This is an analog watch. Probably most, if not all of you, have seen these. It's got a sweep second hand and it doesn't start and stop and start and stop and start and stop. A digital, yeah, it's a little different. You know, we don't know when it gets to 20:59:58 from 20:59:59, it just clicks. It just switches over. This is essentially the nervous system on the right-hand side. The rest of the world, what we take in is on the left-hand side. So I want to try and focus on how do we get from an analog world with all of these inputs that are completely analog and then understand them if we have to turn it into digital signals. Here's what does it. All those things like I mentioned, clicking a mouse, having a dream, falling in love, hating someone, comes down to this particular cell, what we call the neuron. It looks different than most of the cells that we've looked at that are just nice circle and a little nucleus on the inside. It looks very different. This is what they really look like. So the rendition that I just had up there isn't that different. And there are three different, I can generalize it as three different components. We've got what's known as the body, or the soma, S-O-M-A, in the middle, loaded with mitochondria, has a nucleus, I think I can see it right about here. And then all of these extensions coming off of here. And these extensions are referred to as dendrites. And then it kind of tapers down and takes off to the far right-hand side of the board. What it takes off as is the axon. So here's what I had. There's the soma. The dendrites are what's also referred to as a dendritic tree. And then we've got our axon, and then our axon terminals. When we talked about the world of muscle, these little end bulbs, these terminals, these are the things that told the muscles to, hey, contract. They secreted acetyl or acetylcholine. Now, how does it digitize the world? How do we take an analog world that I'm experiencing right now as I look around and smell and hear? You're doing the same thing, and turn it into ons and offs, ones and zeros. That's what we have to try and figure out. Once you've figured that out, you have a much better understanding of how the brain works, peripheral nervous system works, sending a signal to contract a muscle works. This is also an area that, to me, when I took my first neurophysiology course at Wayne State, it was confusing. I think I've said that countless times. I found this topic as confusing as I found many, many others. So, here's what I have to start out with. What really confused me at first was what's called a membrane potential. And the membrane potential is negative 70 millivolts. I was in grad school, and when I heard this, I don't know what that means. What are you talking about? Here's what it comes down to. There are experiments that are done that are called patch clamp experiments, where essentially a little tiny glass tube is pushed through the membrane, and you measure, and this is an important thing, you measure the charge in here, how many ions there are, positive and negative, compared with, relative to, what's on the outside. When you measure what's on the inside versus what's on the outside, the inside is 70 millivolts less than what's on the outside. For me, that was kind of difficult to wrap my head around. But once I started looking into these patch clamp experiments, I started to get it. So here's what we've got. There's all these ions that are inside and outside, but the two most important are sodium and potassium. They both have positive charges. And on the outside, I use a very subjective term, lots of sodium. More sodium on the outside than there is on the inside. On the inside, there's lots of potassium. So I use lots of, but when we look at these two, there's still a negative 70 millivolt difference between the outside and the inside. So how are we going to change that? How do we increase or decrease the resting membrane potential, or the membrane potential? And it's going to be by moving these ions. Move them out, move them in. So there are, as I hesitantly go through this, because as I said, this was difficult for me to understand the first time I learned it. There are essentially two other benchmarks that we want to deal with. There's what's called a threshold, and that's at negative 55 millivolts. You might think, well, what's a threshold mean? We'll get around to that on the next video. And then there is another point which is considered a spike, which is at positive 35 millivolts. So what's happening here is the inside is becoming less and less and less negative. This is all about swapping ions. It's all about moving potassium and sodium. So the reason I'm kind of dragging my feet is that I want to have time. I want to have time in the next video to be able to explain what do these numbers mean, and what happens at the threshold, and what happens at a spike. Now, lastly, well, three things. Right here is the body, or the soma, kind of tapers down to become the axon. This is considered the axon hillock, Hillock. This is where all of that is going to take place. This is the origin of what we call the action potential. Now, the action potential is going to incorporate all of these. It's going to incorporate the movement of ions, and that's what's going to digitize the analog world into how we perceive it. Essentially this is the case. This neuron is either on or it's off. If I look at light and I see different frequencies of light, it's analog, but somehow we have the ability to perceive it, and we perceive it by these neurons that, like I said, are either on or they're off. It's a 1 or a 0. It's binary. So somehow we've got to figure out, how do we do this? How do we convert it over? And then how does the rest of the nervous system work? Here's what we're going to start up with on the next video. It is an action potential. And when you look at this, we have our resting membrane potential. So remember the patch clamp. Patch clamp is in there, and we've got our -70 right here. Our threshold, -55. We'll talk about in the next video what that means to get to that threshold. And then way up here, we have this spike around +35. So this is where we're going next. We're going to use those same numbers, talk about the same cells, and hopefully we can make sense of this all. So I will be back.